So thank you very much to Alain for inviting me to this meeting, and also thank you to you for coming along to listen today. So my lab is interested in the visual system. We're interested in how visual circuits actually work, so how they extract information about the visual scene. And we're also interested in circuit development. So we're interested in how the organisation of circuits emerge, but also how the functional properties of circuits develop. And today I'm going to tell you about some work that my lab's been doing over the last 3 years or so. And what we've been trying to do is to answer a really classic question in visual neuroscience. We've been trying to understand what this structure here, the retina, is actually telling the brain. And also we're trying to understand how the brain actually uses that information. And if there's time at the end, I'll also tell you about the functional significance of layers in the brain, but I'm not sure I'll get to that. But if you're interested, come and ask me at the end. So this is the structure we're interested in, or one of them. So this is a beautiful drawing from Gahal that shows the organisation of cells in the vertebrate retina. And as you all know, the real process of vision begins with these cells. These are the photoreceptors. These are the cells that convert photons of light into the first neural signals in the visual pathway. And these make synapses with these cells, the bipolar cells, which in turn send this information onto the dendrites of retinal ganglion cells. And these vertical pathways in the retina are modulated by lateral connections that are made by horizontal cells and amacrine cells. And it's the axons of the retinal ganglion cells that connect the retina to a whole array of retina recipient targets in the brain. And in vertebrates, there are between 10 and 20 different classes of retinal ganglion cell. It will vary according to which species you happen to be looking at. And historically, these different classes of ganglion cell have been distinguished from one another based on differences in their dendritic morphologies. And I think if you want to understand what it is that the retina is telling the brain, you need to know two things. You need to know what the functional properties of each of these classes of ganglion cell are, i.e. what is each class telling the brain about the visual scene. And you also need to know how the axons of these ganglion cells are distributed to the various, or amongst the various retina recipient targets. So it's certainly not the case that every single target in your brain gets input from every single class of ganglion cell. And so these are exactly the kinds of questions that we've been trying to address. And I think one of the most important jobs of your visual system, one of the most important features that your visual system tells you about is motion, whether it be self-generated as a result of you moving or as a result of objects moving in the environment. So you need to detect motion to cross the road safely. Andy Murray and the rest of us need it for playing sports. Animals in the wild need it for hunting and avoiding predators. And just to illustrate how important motion is, there are people, very unfortunate people, that as a result of stroke, for example, have damage to areas of the brain involved in processing motion. And these people are motion blind. They can't detect motion. And life for these people is really very difficult. So, for example, even trivial things like pouring a glass of water is really very difficult because these people can't see the glass filling up. Even social situations are very difficult for these people. So they can be in a room full of people, they can hear what people are saying, but because they can't see anyone's lips moving, they don't know who's saying what. So detecting motion is really very important, and there are retinal ganglion cell types in the retina of many different species that respond to motion in direction-selective ways. So here's an example of one of those cells. The direction of motion is indicated in degrees around the edge of this polar plot, and you can see that this particular cell responds really well to motion in the forward direction, but not at all well to motion in the opposite direction. So this is a classic definition of a direction-selective cell. And in this particular case, this cell will respond to both light increases, but also to light decreases. So this is an on-off direction-selective ganglion cell. And this table summarises very briefly what we know about these very important class of retinal motion detector. So the type that I just showed you, that on-off direction-selective cell, there are 4 of these, and collectively they respond to the cardinal axes of motion, so up, down, forward and backwards. And the axons of these cells we know project to the lateral geniculate nucleus, but also to the superior colliculus. And purely based on where they project to, We think these cells are probably important for image formation in the case of the LGN, but also for redirecting gaze to new visual targets in the superior colliculus. But this is just based on where they project to. As far as I know, nobody's been able to selectively remove these cells and see what effect it has on behaviour. I'm sure it's probably going to happen in the mouse very soon, though. There's also a second major class of these direction selective cells. These are the on cells, so they just respond to light increases, and there are 3 of these. And you can see that their preferred directions of motion are rotated relative to the cardinal axes. And these retinal ganglion cells send axons to areas of the brain that have a really well-established role in the optokinetic reflex. So this is an image stabilization mechanism. So as you move your head around, you risk smearing the image across the retina, but clearly that doesn't happen. And the reason it doesn't happen is because these cells generate counter-rotations of the eye that exactly compensate for head motion. So as you move your head to the left, these cells will detect retinal slip and generate counter-rotations of the eye to the right. So it's important for keeping the retinal image stable. And lastly, we have this class of cell, an off-direction selective ganglion cell. These respond to upward motion. Again, they send axons to the LGN and the SC. So this is what we do know about these really important cells, but we still don't know an awful lot. And just to illustrate this, this is a montage of a few papers that have come out just in the last few years. I'll spare you the details, but collectively, the field is trying to address 3 questions. So firstly, how many subtypes of retinal motion detector are there? So for example, this subtype is relatively recently identified. How the axons of these retinal motion detectors distributed throughout the brain, we're discovering new projection patterns all the time, seemingly. And number 3, this is the thing that I think we know least about. So we know very little about how local circuits within the brain actually use the information that's provided to them by these direction selective cells. So this is the kind of thing that my lab is interested in. And the model system that we use to address this question is the larval zebrafish, which has already been mentioned by Alain. And even at larval stage of development, the zebrafish retina is organised in exactly the same way as the retina I showed you early on, that drawing from Gahal. The fish has about 12 to 15 different classes of retinal ganglion cell, and these guys will send axons to 10 different retina recipient targets in the larval fish brain. And we work on the largest of these, which is the optic tectum. It sits at the dorsal surface of the midbrain, and it's equivalent to my superior colliculus. So it's important for redirecting gaze to new visual targets. So for a larval fish, the tectum is thought to be really important for catching prey and avoiding predators. And when we started this work, we knew nothing about the functional properties of these different classes of ganglion cell. We certainly had no idea how they projected to these different areas in the brain. In fact, it wasn't even clear that zebrafish even had these retinal motion detectors that I just mentioned. So I can perfectly understand if people in the audience think that we're just reinventing the wheel. Why do we need another system to study motion vision? Mouse is very powerful, for example. And this is where my advert for the zebrafish comes in. So I think zebrafish is a particularly good model system for studying motion vision. And here's why. So even as a baby fish, just a week old or less, they have 3 really robust visually guided behaviours for which the detection of motion is really important. And the first of these is perhaps the most complex of these behaviours. This shows prey catcher. So what you're looking at is a 6-day-old zebrafish larva. It's tracking this paramecium, and it finally gets it. And we know the visual system is important for this because blind fish are terrible. The second movie, you might need to put your glasses on for this one. This shows the optomotor response. And so what you'll see, I'll point it out because it's very small, is a tiny zebrafish larva, again about a week old. The stripes start moving and the fish swims quickly to keep up. Stripes go in the other direction. And the fish swims quickly to keep up. So it's thought that this behaviour for fish is important for maintaining body position in flowing water. If it didn't do this, the fish would constantly being swept downstream. And this last movie shows the optokinetic reflex, again, a week-old fish, and this is exactly the behaviour that you and I have. And you can induce this behaviour by putting the fish inside a drum that has vertical stripes on the inside of it. You rotate the drum and the fish will track the drum by moving its eyes. Okay, so really nice visually evoked behaviours in the fish. Motion vision is important for all of them. The second reason I really like the fish is because at the stages I showed you, the brain is simple. All of those behaviours being generated by a brain that contains 100,000 neurons or less. And the fish is small and it's translucent. And so you can see all of these retinorecipient areas in a single field of view under the microscope. And so just to illustrate that, this is a dorsal view. We're looking down on the head of the fish. Here are the two eyes, here and here. And this is a GFP line in which all the ganglion cell axons are labelled GFP. And just in this single section, you can see 1, 2, 3, 4 arborization fields, 4 retinorecipient targets. And this one's the Tectum, which is what we work on. So what you can do is watch different populations of neurons talking to one another during vision, which is the approach that we like to use to study how the retina communicates with the brain. And lastly, if you're interested in development, and we are, zebrafish is really, I think, quite unique, because all of those behaviours are being generated by a brain that's still very much in the process of assembling itself. And so not only can you use visual stimuli to probe the functional properties of circuits, you can see how those circuits change as circuit development proceeds. And as you'll see later on, there really isn't a simple rule for how retinal ganglion cells develop. And so before I show you the data, I'll just give you an overview of what's happening and when. So the first retinal ganglion cells will begin to send out an axon from the retina around 2 days post-fertilization. They will really innervate the tectum en masse at around 3 days post-fertilization. And then there's a period of retinotectal synapse formation that lasts about 3 days. It's really very rapid. Retinal development is a pretty protracted process. You still have synaptic laminae forming in the retina at 2 weeks and beyond, but the fish is already beginning to hunt at 5 days. So it's behaving despite this really dynamic process of circuit assembly. Okay, so this is the system that we're working on, the retinotectal projection. In fish, all the axons will cross the retina and project to the contralateral tectum. The projection is entirely crossed. Once the axons get there, they'll terminate in one of 4 major layers. This is the major layer, SFGS. About 80% of the axons will terminate here. And then there are 3 sparsely innervated layers, SO, which is the most superficial layer, SGC and SAC are also sparsely innervated, and they're deeper in the tectum. And it's within these layers that the retinal ganglion cell axons will form synapses with a really diverse array of tectal neuron about which we know very little in the fish. Okay, so we were interested in motion vision. We wanted to know how is motion encoded by the whole population of ganglion cells that target the tectum specifically. We're also interested in how the axons of these motion detectors are distributed amongst these different layers. I think this is important to know if down the road you want to understand how a given postsynaptic neuron is integrating all of these information channels coming from the retina. We're interested in how the inputs to the tectum develop in terms of their functional properties and their spatial organisation. And we also want to know how the postsynaptic targets are using that information. And we've developed a technique in fish that allows us to ask these first 3 questions basically just in one shot. And so the way we do that is that we've used a functional imaging approach. And what we've done is to make a genetically encoded and presynaptically targeted reporter of neural activity. And so what we've done is to clone the presynaptic vesicle protein from zebrafish. This protein is called synaptophysin, and we fused it to the genetically encoded calcium sensor GCAMP3. So this targets this calcium sensor to the presynaptic terminal. And this has 2 major advantages. So firstly, it ensures the probe is distributed to the most distal parts of the axon. It's distributed to the synapses, which is the structure we care about. And you're also targeting the probe to where calcium influx into an active neuron is particularly high. So by doing this, you're effectively increasing the sensitivity of the probe. And Ali Walker, who was a really good student in the lab, characterized the probe using hippocampal neurons in culture And the reason we did this is that you can really precisely control the pattern of activity just using extracellular field stimulation. And you can see it has a nice punctate distribution, as you'd expect for a presynaptic protein. And then when Ali field stimulates these cells, we get really nice fluorescence increases. And Ali's done a really detailed characterization of the probe, which we've called SciGCamp3. It works really well. It can report single action potentials. We know it's synaptically localized, has very good dynamic range. If anyone is interested to know more about the properties, I can happily show you after the talk, but in the interest of time, I'm just going to skip this. So what Nicholas Nicolau did, who's a postdoc in the lab, was take this probe and make a transgenic line of zebrafish that expresses it just in the retinal ganglion cells. And here's a dorsal view of the fish, and this semicircle of green, these are all the presynaptic terminals of retinal ganglion cells within the optic tectum. And in this instance, Nicholas has injected a bunch of dye into the retina, and just to show that using this approach, that all of the layers that I told you about are labelled in this transgenic. Then what we've done is to set up a system to provide patterned visual stimulation to our fish while they're sitting on the microscope, and this allows us to record visually evoked activity in the retinal ganglion cell terminals within the tectum. And the stimulus that we use is a really very simple one. It's just bars that drift across the visual field, and then we move the bars through 12 different directions. So we're using this whole setup, this transgenic, this stimulus, to say, okay, how is the direction of motion of the stimulus encoded by the whole population of cells that target the tectum specifically? And just to give you a sense of what that data or an experiment actually looks like, I'm going to show you a movie that Nicholas collected. So what Nicholas did was to show that drifting bar stimulus to the right eye, and he's imaging the contralateral optic tectum, and that's what you're seeing here. So this is the raw data, this is unprocessed. Here are all the axon or presynaptic terminals in SFGS. This is SO, and here are the two deep layers. And this is a delta F over F movie. This shows normalized fluorescence changes. And what we're looking at is a whole tuning experiment. So this is where the fish is watching all 12 directions of motion. And what you can see is really nice stimulus-locked changes, or fluorescence signals in the axons that are for the most part terminating in SFGS. We get a few responses also within the deeper layers down here. And you can see from the clock in the corner that the whole experiment only takes about 6 minutes. So they're very quick to do. Okay, so if we just make a montage of one of these experiments, so each panel shows the response of the retinal ganglion cell axons to the direction shown here in the corner. What you can see from this is very clear direction-dependent changes in the pattern of responses in the axons going to the tectum. So when the bars drift at 90 degrees, so for the fish this would be head to tail, we get really nice responses in the axons terminating in the middle of the tectal neuropil. But as soon as we get any forward component to the motion, now we start to get axons targeting more superficial parts of the tectum lighting up. So this is just qualitative evidence that yes, the zebrafish may indeed have these direction selective motion detectors, but obviously this isn't sufficient. We need to analyse the data in a lot more detail, and this quite busy slide just outlines the steps we go through in order to analyse this data. It's busy, I'll try and make it less tedious, but if I get it out of the way now, you'll know exactly what we're doing in future slides. So the first thing we do is to analyse every single pixel in the movie for one of two types of response. A direction selective response, which is this one, I've already explained this one to you, but we also looked for orientation selective responses. So this would be a pixel that would respond to the bars moving up and down, for example, but not along the orthogonal axis. So this is sensitivity to axial motion. We also found an awful lot of voxels that had responses that looked like this. So they had these really spiky, irregular looking responses. I'm going to call them spikies from now on. These have been identified as a real feature of the developing turtle retina, and we find that they're also a real feature of the fish retina. But I'm going to focus on these two response types. So once we've identified a direction or orientation selective voxel, we then subdivide them into populations or subtypes according to their preferred direction or their preferred orientation. We use a number of different techniques to do that. But once we've identified our functional subtypes, we color code each voxel according to the subtype it belongs to, and then we superimpose that back onto a fluorescence image of the fish that it came from. So what we're doing is we're generating functional maps of individual fish showing what subtypes are in the tectum and where those responses are occurring. Once we've done that, we grab all the fish, all the individual functional maps, and we put them together to make a standard composite map. So this is a much more robust measure of what subtypes target the tectum and where they are. Okay, so finally to the data. So the first subtype we looked for were these classic direction selective cells that actually before we started this thought didn't exist in the fish. It turns out that they do. So we first did these experiments in a 10-day-old zebrafish. So this is when all of those behaviours I showed you are really well established. And so what we're looking at here is a histogram showing the number of direction selective responses per fish and their preferred direction of motion. And we have three subtypes. This really big population here, this corresponds to motion at 254 degrees for the fish. This is tail to head. And two much smaller populations that respond to up and downward motion. And if we generate polar plot to these populations, you can actually see that they're quite broadly tuned, i.e. this population will respond to a range of different directions rather than having a very discrete preferred direction. But collectively, these three populations really nicely cover all directions, and there's very little functional overlap between these subtypes. It's almost like they're tiling direction space. What's really striking about these cells, if you're interested in development, is how little they change during development. So we did these experiments again in a 3-day-old fish, and this is really during the phase when axons are still just reaching the tectum. And you can see that these responses look exactly the same as they do at 10 days. So direction selective circuits in the zebrafish retina wire up extremely quickly, and they wire up extremely accurately. There's no functional refinement over time. So for the fish, presumably there's something very important about these direction selective cell types. And here's how the preferred directions of motion corresponded to the body axis of the fish. This is up, down, these are the small populations, and this is the major population. This is these guys here. So I think there are two particularly interesting things about these cells that we've identified in the fish. The first is, in terms of their number and their preferred direction, they're uncannily similar to the on-direction selective cells that you find in mouse, for example. And if you remember back to that table I showed you, these are the cells that drive the optokinetic reflex. What's interesting is that you can ablate the tectum in a fish and the optokinetic reflex is entirely intact. So one idea is that either the tectum is involved in the OKR, which is just a subtle role. The alternative interpretation, and this is one I think is much more and potentially much more interesting, is that these cells do drive the optokinetic reflex in the fish, but at some other target. The implication being that these cells, when they project to the tectum, are being reused for some other purpose. And the reason I think this is interesting is that you can use the same type of information, the same channel of information coming from the retina, and reuse it multiple times depending on where it goes to in the brain. I think this is something we'd really like to address more. The second interesting thing is that we found no cells in the retina that were selective for this direction. And we were surprised at this. We thought if there's one thing the fish wants to know about, it's stuff coming towards it. And we just didn't find any of these cell types in the retina. And I'll come back to this later on. So the second subtype we looked for were the orientation selective responses. And at 3 days, there was only one subtype. And these responded to vertical axis motion. But at 10 days, there were 3, possibly 4 subtypes. 3 clustered around the vertical axis and 1 that corresponds to horizontal axis motion. So for the most part, these these orientation selective responses emerge later in development than the direction selective ones do. Okay, so we've identified these two broad subtypes, direction and orientation selective responses. Where are they going to in the zebrafish tectum? So to look at this, we made our functional maps. And what we've done in this case is to lump all the direction selective subtypes together and color-coded them magenta. We've lumped all the orientation selective subtypes together and color-coded them green. And these are the spiky turtle-like responses. They're colored in cyan. And if we first look at the direction selective responses, no matter what stage we look at, whether it's a really young tectum or a very mature one, no matter what stage we look at, they always target this really nice, neat lamina just within the superficial parts of SFGS. So everything about these direction selective cells seems completely invariant, from their functional properties to the way these axons innervate their target. Now if we look at the orientation selective responses, at 10 days these are also organised into lamina, but they're organised into two lamina, one in the middle of SFGS and one in the deeper part of SFGS. And if you look at this line plot, so that this line shows the distribution of the direction selective cells, that's these one, and the green one shows the distribution of the orientation selective cells. You can see one lamina, which is this one, second lamina, which is this one. What's interesting is that there's virtually no overlap between these two subtypes. So at least with respect to these cell types, axons are segregating within the target according to the type of information they convey about the visual scene. So there's this very kind of nice, simple logic to the organisation of these axons in the target. But this laminar organisation of orientation selective responses, it takes time to emerge. It's not like the direction selective cells. So you can see here you have this kind of patch just in the posterior part of the tectum, then you have this diffuse haze, and slowly you refine into this lamina over time. So I think this reinforces the idea that there are real developmental differences between these two classes of cells. Direction selective cells, invariant, nothing changes. Orientation selective cells emerge later and they refine their organisation slowly over time. Okay, this last subtype, these are the turtle-like responses, these are the spiky-looking responses. These are actually the dominant class early in development. They're all over the tectum, but then you gradually lose them over time. And to me, these are perhaps the most interesting ones. We'd really like to know what happens to these. What happens to these cells? The other interesting thing is, yes, the fish is seeing during this period, yes, its visual system is being used to drive behaviour, but the input the brain is getting from the retina is pretty noisy. It's certainly in the process of refining. So I think the tectum of the fish is a really nice place to see how postsynaptic circuits deal with this developmental noise. This is something we're also looking at. How am I doing for time? Okay. So the one thing we couldn't understand using our population functional imaging is how refinement is actually taking place in the tectum. So remember I showed you the orientation selective voxels look like this early and then they refine into this later on. Because we can't see the structure of individual axons, we don't really know what's happening. So this refinement could be happening either because axons are changing their functional properties, so they're either losing orientation selectivity or gaining it. The alternative is that the axons themselves are physically refining. Early on they explore multiple lamina and later on they refine over time. So to address this question, what we did was to label a single retinal ganglion cell in vivo with our functional indicator. And because we've got only a single cell labelled, we don't have that blur of fluorescence from other labelled cells. We can get the morphology and function of the cell at the same time. And so here's a cell we labelled. So this is the axon just as it's arriving in the tectum. You can see it has a very simple arbor, but then it grows massively by 7 days post-fertilisation. But then if we rotate this and look in the plane of the layers, you can see that no matter what stage we look at, this axon stays perfectly planar. So it's never exploring multiple layers in the tectum. And then when we do functional imaging on this cell, if we did it at 3 days and we showed that cell the drifting bar stimulus, it just didn't care. We got no response from it at all. But then if we switch the room lights on, we got this really nice response. So a full field flash, you can get the cell to respond. So it is beginning to plug into the retinal circuitry. If we then do the experiments for 5 days, now we get these beautiful responses to the drifting bar stimulus, but it responds really well to every single direction. So it's not tuned in any way. If we then look at 7 days, this is the same cell, now it's much more selective. Now the cell is only responding to vertical axis motion. So it goes from not responding at all, or only responding to full field flashes, to being very nicely orientation tuned. So we think that the functional refinement we see over time is not because axons are physically changing, it's because they're changing their functional properties. Again, reinforcing the idea that the zebrafish brain is getting refining, noisy input from the retina. Okay. So that was done, that was the presynaptic work. Now we know how the retinal ganglion cells targeting the tectum, we know how as a population they encode this drifting bar stimulus. This gave us a really nice opportunity for saying, all right, well, what do the postsynaptic cells do with that information? And the approach we used was really very simple. We did exactly the same kinds of experiments we did on the axons, but now on the postsynaptic neurons. We looked for populations and we just compared the two structures. We compared the retina to the tectum and just to see what would change. And so here's a movie of the tectoral cell responses. So here are all the tectal cell bodies. This is the neuropil where all the axons are coming in. And this is a tuning experiment. Again, fish is watching that drifting bar stimulus, and you can see really nice responses in the tectal neurons. And this is work that's been done by Paul Hunter in the lab, who's a postdoc. Okay, so the first thing Paul did was look for orientation selective cells. So this is a single functional map from a single fish. Paul identified orientation selective cells and color-coded them according to preferred orientation. And you can see, yes, they do exist, and they're broadly distributed throughout the tectum. And when Paul looked for functional subtypes, he found exactly the same functional subtypes that we found in the retina. The simplest interpretation is that the orientation selective tectal cells are getting input from the orientation selective retinal ganglion cells and just reading that information out. When we looked, or I'd say when Paul looked for the direction selective tectal cells, it got much more interesting and strange to us. So again, direction selective tectal cells do exist. They're broadly distributed throughout the tectum. And when Paul looked for functional subtypes, he found tectal cells that were selective for upward motion, downward motion, and this big population forward motion. And remember, these are exactly the same preferences that we saw coming from the retina. But now, Paul finds this really sizable population in the tectum that is now selective for motion going backwards. And this was a response type I was at pains to point out was not present in the retina. So this selectivity for posterior motion is generated de novo within the tectum. It's an emergent property of tectal circuits. Somehow the tectum is generating this from inputs in the retina that are not tuned in this way. So just a brief summary. So this is the retinal representation of motion direction. It's represented by these three subtypes, and they have this kind of triangular arrangement to their preferred directions. But then when you move to the tectum, you have 4 direction selective subtypes, and their preferred directions are in a cardinal arrangement. So we have 2 really interesting questions, well, we think they're interesting, which is to try and understand why the tectum does it, but also how the tectum does it. We have no idea why the tectum does it. We have a few models for how the tectum does it. If anyone's interested, I'll go through them. But yeah, so, oh yes, so the last thing Paul did was he was really interested to know whether there was any spatial organization within the tectum. Exactly like we saw in the inputs, everything was highly regular and laminar. Paul wanted to know if there was any functional segregation within the tectum. The poor guy spent about a year looking, and there isn't any. So this is just three-dimensional functional maps which Paul generated. So the retinal ganglion cell axons are shown in grey, and here are all the direction selective cells colour-coded according to subtype, and same for orientation selective cells. So there's no functional segregation within the Tectum according to subtype, at least at the level of cell bodies. I think we'll probably find it if we look in the neuropil, but not at the cell bodies. Okay, so just to summarise, we have 3 types of direction selective cell and 4 types of orientation selective cell that project from the retina to the Tectum. These inputs to the Tectum are arranged into separate layers, and these 2 subtypes develop differently. Everything about direction selectivity seems invariant, whereas orientation selectivity, particularly their functional organisation, refines over time. So I don't think retinal ganglion cells can be treated as one simple class. They're different. And lastly, this was our most striking result, is how the Tectum transforms the retinal representation of motion direction. This is something that I think we're going to be studying for at least a few years. Hopefully I can give this talk again, tell you how it does it. Okay, I'm just going to finish by saying thank you to Paul, who did all the tectal cell work, Nicholas, who did all the retinal ganglion cell work, Ali characterised the probe, and this is Fatima, she just joined the lab. Andrew Lowe was a really fantastic collaborator at King's, helped us with the analysis. Juan gave us reagents. Chi-Min Chen, he was a fantastic resource for the Zebrafish community. Sadly, we've lost him. And also thanks to Koichi Kawakami. So thank you for your attention. I'll happily take some questions.